Today, we now know that the majority of genomes in complex organisms are transcribed not into proteins but into non-coding RNAs. A non-coding RNA is a functional RNA molecule that is transcribed from DNA but not translated into protein. There are three main classes of short non-coding RNAs. MicroRNA is one of them. MicroRNAs represent approximately 4% of the genes in the human genome and play an essential role in gene silencing and post-transcriptional regulation of gene expression in plants and animals. MicroRNAs are approximately 22 nucleotides long and are derived from transcribed hairpin structures. siRNA function in a similar way to microRNA in that they mediate post-transcriptional gene silencing. siRNAs are 20 to 23 nucleotides long and are synthesized from double-stranded RNAs such as hairpins with a long, perfect stem or sense-antisense transcript hybrid. The most significant difference between microRNAs and siRNAs is whether they silence their own expression. Almost all siRNAs are capable of silencing the same locus from which they are derived. piRNAs are typically 24 to 30 nucleotides in length and are involved in chromatin regulation and the silencing of transposon activity.
In animals, microRNA is synthesized from primary microRNA in two stages by the action of two RNase 3 type proteins, drosha in the nucleus and dicer in the cytoplasm. At the beginning of transcription in the nucleus, single-strand primary microRNA gets a cap and a poly-A tail. The cap and poly-A tail can be spliced by the dicer enzyme in further processing and pre-microRNA is formed. After these two steps, pre-microRNA is exported from the nucleus to the cytosol with the assist of exportin 5 and RAN-GTP. For plants, they have a dicer-like 1 enzyme which has the function of both drosha and dicer activities. Because these enzymes are located in the nucleus, microRNA have already formed the duplex before it's exported to the cytosol. Then, exportin 5 homolog hasty transports the microRNA duplex. MicroRNA can bind to part of the target messenger RNA by complementary base pairs. According to this process, microRNA inhibits ribosomes binding to messenger RNA and later influences the elongation. As this destruction happens, the poly-A tail of messenger RNA degrades and causes the final death of messenger RNA.